Now, we are going to learn a few tips and tricks to improve your experience with XRNA. First, we have several hotkey combinations which act as shortcuts for UI interactions. To use these hotkey combinations, you must first click somewhere within the XRNA program. If you attempt to use these hotkeys before clicking into XRNA, your browser will attempt to interpret these commands in ways which have nothing to do with XRNA. Number 1. Ctrl-O launches a menu for opening files inside XRNA. Number 2. Ctrl-S launches a menu for saving files outside XRNA. Number 3. Ctrl-Z undoes an action. These can include click and drag actions, edit actions, format actions, and annotate actions. Here, we click and drag a nucleotide to demonstrate the ability to undo. Number 4. Ctrl-Y redoes an action which has been undone. This command can also be performed using Ctrl-Shift-Z. Please take note, you can also undo and redo using these two buttons. Number 5. Ctrl-0 resets the viewport. None of these hotkey combinations require you to have a certain tab open. You can use them at any time to accelerate your use of XRNA. One other fun trick is the nucleotide freezing feature. Click a nucleotide using your middle mouse button or scroll wheel to freeze a nucleotide. Frozen nucleotides will appear to fade in and out of existence. Frozen nucleotides are ignored by actions such as click and drag, editing, formatting, and annotating. You can unfreeze nucleotides in the same way you froze them, by clicking the middle mouse button. Frozen nucleotides can also tweak the behavior of constraints, such as the RNA single-strand constraint. Also note that you can freeze whole substructures at once using the appropriate constraint. One final trick which may improve your experience is the right-click and drag function. Let's zoom in to see it in action. When you are using the Edit tab, the Format tab, or the Annotate tab, you can skip manual selection of a constraint. Instead, simply right-click and drag across as few nucleotides as possible to select your desired substructure. In response, XRNA attempts to infer your desired constraint. Then it populates a left-hand menu. XRNA accomplishes this by examining the nucleotides which the user clicked and dragged over. Then, it iterates from the most specific constraint to the most broad, attempting to match all dragged-over nucleotides to a single constraint. Therefore, the more nucleotides the user selects, the broader a constraint XRNA auto-detects. Now we will demo a few examples. Dragging over a non-base paired nucleotide results in the single nucleotide constraint. Dragging over two mutually base paired nucleotides results in the single base pair constraint. Dragging over a contiguous run of base pairs results in the single helix constraint. Dragging over a large selection of unrelated base pairs results in the RNA molecule constraint. Thank you for watching. See our other videos to learn more.